Hi everyone, this is Team Beyond the Tent and our objective is improving urinalysis for refugee healthcare. We are a team of 4, Scott Piper, Janasa Ramprasad, Frugal Sonyal and Vidya Jose. The problem in hand, refugee crisis, is at its highest since World War II with close to 26 million people suffering worldwide. This amounts to 1% of the globe's population and sadly 40% of them are children. The countries that host 85% of this vulnerable group are themselves resource limited. Now among the multitude of problems that they face, health issues get the center stage. Though their general health might have been good enough, it worsens due to the living conditions in refugee camps. Poor ventilation and sanitation, water leaks, pest infestation, overcrowding are few. They are severely malnourished and their camps often form breeding grounds for various infectious diseases and STDs. Refugees are more likely to have poorer mental health than the locals as well, with higher rates of PTSD and depression. Hence, it is all the more significant to come up with frugal solutions in this space of refugee healthcare. The 3 solution spaces we charted out that are crucial are 1. Basic needs, 2. Diagnosis, and 3. Treatment. Basic needs include an overall improvement in their living conditions, providing good nutrition, clean drinking water, sanitation facilities, and an access to primary health care. Since they live in crammed up spaces together and the chances of diseases among them going unchecked and untreated high, the consequence of a prospective outbreak would be severe. Next is obviously to cover the options of treatment available, be it for cancer, AIDS, TB or other illnesses. We initially didn't have our head sorted for just catering to one problem and its solution, since, as would be clear by now, the refugee healthcare system has a lot to get fixed.
The original idea board did not come with a specific problem. We spent the majority of the class trying to navigate and narrow down the problem question. We reached out to Jude Alava, an MD student at Stanford, and learning about his past work and experiences with refugees in Syria helped us understand the differences in living conditions at different camps and life at urban centers. Their main challenges were continuity of care, access to specialized care, and language barriers. Initially, we were inclined towards looking at telehealth ways or a software-based system to connect doctors from all around the world to a centralized patient history system, but we were not very sure about its efficacy and feasibility. We also understood that a large number of applications, websites, and tools already exist in this niche, and we will have to come up with something truly different to make us stand out and practical. We also discussed in length about language and cultural barriers and inclusion of maybe a questionnaire or DSM-5 like manual. Changing policies is beyond the scope of this project, but we would definitely incline towards working at something which not only looks good on paper but also eases the quality of life. We realized during this project that refugee health is something very complex and the socio-political and geographical factors further entangle it. Lack of sufficient nutritional food, poor living conditions, lack of primary and preventive health care are major issues for malnutrition, morbidity, and mortality. Further, the team reached out to Laura Kwang, a PhD student at Stanford. Her work in Rohingya refugee camps, which is one of the largest refugee camps in the world, housing more than 1 million refugees, helped us understand their living conditions in a much better way. We realized that they are not allowed to leave these camps and they are not even integrated into an urban center. Since most of these refugees come from low-income farming backgrounds, their health issues are substantially different from that of Syrian refugees. Lack of basic resources helped us pivot towards looking at something paper-based that would be a lot more inclusive and accessible. In Bangladesh, unlike Turkey, the majority of refugees still live in very primitive camps that lack basic medical supplies. One struggle that Laura personally encountered and made us realize was the lack of a dedicated lab space in the camps, meaning that any tests for water quality or diseases that required microscopy, incubation, or centrifugation, or any other complex processes were not possible. This led to the team looking at paper-based diagnostic tests.
One of the most common paper diagnostic tools, and the one that we decided to analyze, is Urinalysis dipstick test. A typical urinalysis dipstick contains 10 testing squares, each with a different colorimetric test. These include specific gravity, pH, proteins, leukocytes, nitrites, blood, ketones, glucose, bilirubin, and urobilinogen. The procedure to use these tests is simply to dip them into a urine sample, shake them off, and wait 30 to 60 seconds before reading the color against the provided key. These tests can be bought commercially for around 30 cents in the US. In higher resource areas, these are used as a screening tool to detect changes in the analytes that may point to various underlying conditions. However, in a setting such as the camps in Bangladesh, there are limited resources to run the necessary follow-up test, which may require blood work, microscopy, or culturing. Thus, in these settings, the dipsticks may take on more of a diagnostic role or help healthcare providers know where to best use their resources. After studying this technology, we wanted to find a way to improve its cost-effectiveness in refugee camps. Our guiding questions for this process were which tests are most important for our audience, and do we need to use all 10 tests on each patient? This led us to the first part of our solution, which was identifying the most important tests for refugee healthcare. Our goal became to narrow down the strip from 10 tests to 4 tests on a simplified strip. To help our decision process, we identified 4 key criteria, which were the relevance to a refugee camp setting, the ability to identify a specific diagnosis or cause for a certain test result, the simplicity of the result readout for healthcare workers with minimal training, and the stability of the analyte in a urine sample. We'll start with a quick overview of the tests that we rejected. A more complete analysis of each test is available on our Notion. First up, we have specific gravity, which measures the amount of solutes that are dissolved in the urine. There are a large variety of conditions that can cause elevated or decreased levels of dissolved solutes. Additionally, long periods of monitoring this parameter are often necessary, making it not as useful in our setting. Next is the test for bilirubin, which is a product from the liver that is normally filtered out of the urine. This test can help you to get an idea of how well a patient's liver is functioning. We rejected this test because bilirubin can break down if the urine sample is exposed to light. The color change is from one shade of yellow to another, so it's hard to read, and it is redundant with the urobilinogen test, which also investigates liver function. Next, we analyze the pH test. Normal urine is slightly acidic, so any change from this base value in either direction can indicate a problem. However, urinary pH can change due to many benign factors, such as diet and time of day. Urea in the urine can also convert to ammonia, which provides a misleading alkaline result. For these reasons, the pH test is not as good of a candidate for a refugee camp setting. Leukocyte esterase was the next test we analyzed. This enzyme is produced at increased levels in the presence of an infection, and it is eventually excreted from the body through the urinary system. As we will talk about in a few slides, the nitrites test also detects infections, and the leukocyte test has been shown in literature to have a very low positive predictive value, which could lead to overtreatment, which would waste valuable resources in a refugee camp setting. Next, we looked at the test for blood, which is another one that we had to reject. There are 2 types of hematuria, or blood in the urine. Gross hematuria is when large amounts of blood are present, which changes the color of urine to a dark brown. The dipstick tests for microscopic hematuria, where the amount of blood is not detectable with the naked eye. There is a very long list of illnesses that can cause microscopic hematuria, and many follow-up tests that are not available in these camps are required to get a specific diagnosis. Because of this ambiguity, the blood test had to be rejected. Finally, we rejected the protein test. In a normal sample, there may be trace amounts of protein in the urine, so this dipstick test looks for levels elevated beyond a trace amount. Again, similar to hematuria, proteinuria has many potential causes. However, we did note that monitoring protein levels in the urine may be helpful in managing diabetes. Now, we will cover more in depth the 4 tests that we determined were most important to include, the first of which is nitrites. During an infection, gram-negative bacteria, such as E. coli, convert nitrates in the body into nitrites. Testing for nitrates in the urine thus acts as a testing for a pseudomarker of the infection itself. As we learned from Laura and the research done by the organization ELRA, one of the largest problems in the camps is the inability to test whether their water sanitation systems are actually effective or not. Until a solution is found to verify the effectiveness of their water treatment, monitoring for E. coli infections will be imperative to maintaining the refugees' health. This is one of the simplest ways to test for an infection without advanced equipment. The color change for this test is to various shades of increasingly darker pinks. The chemical reaction for the test is called the grease reaction, and it takes place in 2 steps. First, the nitrite reacts with sulfanilamide, which is actually an antibiotic drug, to form an intermediate, which then reacts with NAD to produce a dye. Another test we included was for urobilinogen. This compound is produced as bilirubin is digested. Under normal conditions, around 1 mg per deciliter is excreted in the urine. If levels higher than this are detected, the cause can be directly traced back to liver issues, commonly either cirrhosis or hepatitis. Specifically, in newborns, elevated urobilinogen points to a hemolytic disease. If decreased levels are found from the baseline, there may be blockage in the biliary ducts, meaning not enough bilirubin is being secreted. Although bilirubin could be used to analyze liver function, the readout for bilirubin is not nearly as helpful. As you can see in the diagram here, bilirubin tests can detect relative levels while urobilinogen results are quantified. Additionally, the color change for the urobilinogen test is more shades of red or pink, which is easier for an untrained eye to differentiate than the shades of yellow that are used in the bilirubin test. The chemical reaction for this test relies on the key substrate of p-dimethylaminobenzyaldehyde, as shown here. Next, we decided to include the test for glucose. In normal kidney function, glucose is filtered out of the blood and then reabsorbed to limit any carbohydrate waste. In the case of hyperglycemia, the amount filtered out of the blood exceeds how much can be reabsorbed. This excess is then excreted in the urine. The CDC has acknowledged that this test can be used in helping to identify asymptomatic diabetes. If a test for glucose comes back with elevated levels, it can help guide what other tests need to be done, such as the ketone test, which we'll talk about next, or the protein test, like I mentioned earlier. The color change can be seen here from a light turquoise to increasingly darker shades of red or brown. The chemistry for this test follows a 2-part reaction. First, glucose is oxidized into gluconic acid and hydrogen peroxide. The hydrogen peroxide then reacts with a dye in the presence of horseradish peroxidase to cause a color change. The final test we found to be important was the ketone test. Ketones are produced in the body when fats are digested instead of sugar. One of the main reasons this may happen is during starvation conditions when the body must use fat stores for energy. Another cause of ketonuria is diabetic ketoacidosis, which occurs when the body doesn't have enough insulin. Diabetic ketoacidosis causes dehydration, acidic blood, vomiting, abdominal pain, low blood pressure, and eventually a coma and death. This test, combined with the glucose test, form a pair that is critical in monitoring diabetes mellitus. The American Diabetes Association has listed ketonuria test strips as essential equipment in managing diabetes in conflict settings. The color change from this test is a yellow to increasingly darker shades of purple. The chemistry is based on a reaction called Rothera's test. The reaction is specific to acetoacetate, which reacts with sodium nitroprusside to change colors. So, nitrates, urobilinogen, glucose, and ketones are the 4 most important tests for a refugee setting based on our research. Next, Projal will tell you about the second part of our solution.
Hello everyone, my name is Prajal Sanyal and I will be going over the second part of our solution. Solution part 2, rethinking the strip. While focusing on our solution, we thought of a second addition to the solution could be possible. The sample collection for diagnostic tests in refugee camp is a problem, as there is a lack of trained professional doctors in the refugee camp. So we thought of using a device that would make the test more patient oriented or less focused on personnel. Therefore, we thought of using a modular tube. This tube would have a ladder shaped slit on the upper portion of its body. The strip slit will house the test strip, and this tube can be given to a patient. The patient can pee directly inside the tube up to a level marked by guidelines on the outer body of the tube. After the patient is done filling the tube till the marked line, the tube can be shut by the cap that is attached to its body and then turned upside down. Allowing it to stand for a few minutes, it can be turned upright again. This allows for the test strip to be submerged in the urine sample for a short period of time without degrading it too much so that the color leaks into the sample itself. After the test strip has sufficient amount of time to be submerged in the urine sample, we can observe it by turning the tube upright again. Any color changes would depend on the concentration of the analyte that we are looking for in the sample. This makes it easier for the patients to test themselves. This also eliminates the need of a personnel to dip each strip into individual urine samples, which could also be very unsanitary in places like big refugee camps. Moving on to the future part of our work. Even after this class ends, we hope to connect with a doctor who is already working in refugee camps and understand with them the tests that are most important in those particular environmental conditions or lifestyle conditions, as literature research would answer only so many questions for us. This way, we will also learn which tests are most important and which test we should keep, as minimizing the number of tests also minimizes the cost of production of the strip. Then we would also know if the doctor or the people over there, the patients themselves, think if the modular plastic tube piece is something that is necessary for them, that's something they would use. And then inclusion of the tube would depend on what their feedback is. Next, we would look into the manufacturers of the strips themselves. We would reach out to them and ask them if they are ready to reinvent their production just a little bit so they can produce tinier strips with less test for at lesser cost, so that this can be offered to refugee camps. Rest of the information for our project is available on our Notion page. It has been laid out detailed under individual tabs. For any more information and queries regarding our project, you can reach out to our team contact Scott Piper at his email given in the slide. Thank you for your time and attention.